Good day, everyone. So today I'm going to be presenting on the evaluation of microbial population in a smallholder oil palm plantation. I am Anisia Benedict Bakar 64643, and this is my final year project presentation. These are the things that I'm going to be talking about throughout the whole presentation. Moving on to the introduction. So peatlands only cover 3% of the whole globe, which is very little if you ask me. And the coverage of virgin peatlands are progressively declining because these lands are constantly being converted into agricultural lands. My study is going to be focusing on oil palm plantation. And oil palm species, the most commonly planted oil palm species is the Elaeis guineensis. And this industry is the backbone in the development of agriculture and economy in Malaysia. As I said before, it contributes massively to our gross domestic product and gross national income. And as of 2020, around 300 smallholders are registered in Malaysia. Moving on to the problem statement. So there are two problem statements in my study, which are peatland in Malaysia is still understudied, and also the effect of land conversion on the microbial community is not fully determined yet. So these are the objectives of my study. Moving on to the literature review. So the first point is soil microorganisms and their roles in the peat soil environment. So here I have listed microorganisms that are responsible for the processes that happen in the soil. Decomposers are basically microorganisms that are able to break down either tough cellulose, lignin, or chitin. The carbon cycle are microorganisms that are able to produce exoenzymes such as Rubisco to aid in the carbon cycle. And the nitrogen cycle involves three steps, which are nitrogen fixation, nitrification, and also denitrification. There are two important factors that affect the diversity of microbial community in the peatlands, and they are soil pH and soil water content. These two factors affect the composition of the soil, also the microbial population and community in the soil. So the peatland ecosystem are generally divided into two, which are fen and bog peatland, and the differences can be seen in this table, where fen peatland has higher nutrient content and bog peatland is slightly more acidic compared to fen peatland, although both of them are acidic. And the dominating vegetation in fen peatland is carex species, while in bog peatland it is sphagnum mosses. So there are two groups of smallholders in Malaysia, which are independent and organized smallholders. So independent, as the name states, they are independent and they manage their land individually without support of government. And the organized smallholders are supported by government-led agencies such as FELDA, SALCRA, and so on and so forth. So moving on to the methodologies, the details of the sampling site can be seen in this table. I have performed the sampling in Ulu Sebau, Bintulu, Sarawak on April 26 at 5 p.m. And the sampling method that I've chosen is soil surface sampling. So as 3 replicates of 50 grams of peat soil were collected in 3 locations. One is under the leaf canopy. The second one is the spaces between the rows of oil palm trees. And also the third location is taken from a random spot within 2-meter radius from the palm tree. So the determination of soil properties were done in situ and also in the lab. So properties such as soil moisture and color were determined in situ because they can be observed. And for the lab analysis, the pH of soil were analyzed by using the standard procedures. In my study, I used the 1:2 soil to water ratio. So the next one is the enumeration of total bacterial viable count. So I used the spread that method on nutrient agar, and the final dilution of 10 to the power of -1 and -2 were chosen to get just enough colonies on the plate. And these plates are incubated for 16 hours at 37 degrees Celsius. And individual colonies formed on the plates were counted manually. The average number of bacterial colonies formed on these agar plates are then calculated to be used in the next step, which is the calculation of colony forming unit CFU. So this is the formula that I am using. So I used it to find the colony forming unit per milliliter of the sample. Moving on to the results in discussion. So the first result is on the pit soil properties. The soil moisture in both pit samples are high. Water grizzled out of my grasp as I squeeze it. The color of pit soil though is different in the two locations, which is dark brown in location 1 and blackish in location 2. All right, moving on to the discussion. So pit soils generally have a dark brown to blackish color due to their high content of organic matter, and water can make the soil appear darker temporarily by increasing light absorption. So I think this applies to the different coloration in the pit soil in location 1 and location 2. So it shows that location 2 has slightly higher water content compared to location 1. And soil moisture is said to be corresponding positively to the composition of certain microbes in the area, such as prokaryotes and protists. So a reduction in microbial population can occur in a condition where the moisture content is too high or too low. So because if the water saturation is too high, the population of aerobic bacteria is threatened because they cannot perform aerobic processes in the ground. Moving on to the next discussion, which is the comparison of bacterial communities between natural forests and converted peatlands. So I have incorporated the study done by Kusai and Ayop in 2020 that shows the differences in microbial community among natural peat swamp forests and peatlands that have been converted into oil palm plantation. And their study have found out that the common phyla in all sampling locations, which are the peat swamp and the oil palm plantations of different ages, are Actinobacteria, Firmicutes, and also Proteobacteria. So Proteobacteria are usually found in places or environments that have high nutrient content, such as carbon. And Firmicutes are known as plant biomass decomposers. So they are found in places with high amount of plant residues. So the comparison of microbial population between natural forest and oil palm plantation. The quality of soil in natural forest is high because of the rhizomicrobiome in the area is not disrupted, and they can perform biological processes efficiently. In oil palm plantations, they are constantly being treated with fertilizers, and this causes the soil to be too saturated. And also limited soil carbon threatens the population of rhizomicrobiome in the environment. And microbial population are generally higher in undisturbed natural forests compared to there is in oil palm plantations. So in conclusion, microbial population are greatly affected by physiochemical properties in the soil, such as soil water content, soil pH, soil carbon, and so on and so forth. And in my study, there is a lack of significant difference in the pH value. So there is no telling that the pH value affects the population, but I can confirm you that it affects the community of microorganisms in the soil. Proteobacteria, Firmicutes, and Actinobacteria are three of the most common phyla that can be found in both natural peat forests and also oil palm plantation. And the population of microorganisms in cultivated lands such as oil palm plantation are found to be generally lower compared to undisturbed natural forests. So that's all for my presentation today. Thank you for your attention. Have a nice day.